All right, can everyone see my screen okay? Just give me a thumbs up if it looks good still. All right, awesome. Everyone, thanks for joining. We'll give it about 1 minute here and then we will get going. All right, so to keep it on track, I'm just gonna get us started here. It is 101. People are still probably gonna be trickling in, but I can at least get through some of my housekeeping. So hello and welcome to the Joint Invasive Mussel Collaborative and Great Lakes HABs Collaborative Webinar. We have two excellent speakers today who've agreed to share their work with us. Presenters Anna Baghold with the Cooperative Institute for Great Lakes Research and Vincent Deneff with the University of Michigan will be sharing their work regarding Quagga mussel food choice at the HAB buffet. So my name is Nicole Angel. I'm joined by my colleagues Connor Ressler, Sam Tank, and Teresa Greninger with the Great Lakes Commission. And on behalf of the IMC and HABs Collaborative, we just want to thank you for participating in today's webinar. So before I turn the presentation over to our presenters, I just wanted to provide some background about each of the collaboratives that have organized this webinar today. So the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for the management and control of invasive Dreissenid mussels in order to protect and produce measurable ecological, economic benefits. The Invasive Mussel Collaborative published a management strategy in 2018 to reduce invasive mussels and their negative impacts. So to make progress on these goals, the collaborative established 4 work groups: the research work group, the Dreissenid toxicity testing work group, the coastal site priorities work group, and most recently, the planning and implementation work group. So I'll briefly highlight each of these work groups in the next couple slides. So the research work group was charged with coordinating existing research and developing targeted plans and projects that address Great Lakes management priorities. About 2 years ago, the work group released a mapping tool called the Dreissenid Project Coordination Mapper, which allows researchers and managers to share their work and collaborate with others for the advancement and protection of waterways from invasive mussels in the Great Lakes and beyond. So the work group is currently soliciting additional research and applied control projects, which can be submitted directly through the mapper or using that bit.ly URL at the top of the screen there. The next work group is the Dreissenid toxicity testing work group, which was charged with reviewing the strengths and weaknesses of mussel toxicity control methods used across various labs and to identify and make recommendations to further refine and standardize these methods. So the studies reviewed for this work have been compiled into a table that provides a searchable collection of literature for Dreissenid laboratory toxicity and open water control testing methods. And this is available on the Invasive Mussel Collaborative website. And additionally, a manuscript detailing the recommendations made can be accessed by the bit.ly URL on the screen. And then the Coastal Site Priorities Work Group was charged with reviewing information on where Dreissenids are impacting Great Lakes resources, evaluating sites based on this information, and developing and implementing a system for prioritizing sites for potential management activities. The work group released an interactive dashboard and map that displays nearshore data from the Coastal Site Priorities Index, and the tool called the Great Lakes Experimental Mussel Site Suppression Screening Tool, kind of a mouthful, but you can otherwise refer to it as GLEMPST. This tool features a dashboard and associated indices which are intended to inform future applied Dreissenid research and management activities by aiding site prioritization. And the tool, again, can be accessed by that bit.ly URL on the screen. And then finally, the planning and implementation work group is the most recent of the work groups, and it was charged with developing a research and management roadmap for site and regional scale mussel control tools, in addition to identifying finding priorities and funding priorities and remaining knowledge gaps to guide future Dreissenid control activities. So this work group is sort of building on the products and outcomes of the other 3 work groups to develop a research and management roadmap for site-level Dreissenid mussel control. So be on the lookout for more exciting progress from this work group over the next coming years. All right, and then on to the Great Lakes HABs Collaborative. The HABs Collaborative was launched in 2015 by the Great Lakes Commission and the U.S. Geological Survey Great Lakes Science Center, and is working to establish a common agenda on the science and management needs to help the region work together to prevent and manage HABs. The goal of the Great Lakes HAB Collaborative is to improve communication among scientists and between scientists and decision makers on issues related to harmful algal blooms in the Great Lakes. So the HABs Collaborative has a research mapper, which is an app to share regional research projects to increase collaboration among researchers and water managers working to investigate and address HABs in the Great Lakes Basin. If you have anything to add to this mapper, please consider adding. You can find more information using the QR code on the screen or on the HABs Collaborative webpage. So today's webinar is part of a communication network facilitated by the collaboratives. The purpose of these webinars is to really facilitate learning and information sharing on common topics of interest. So the webinar will be recorded and archived along with past webinars on the Invasive Mussel Collaborative and HAB Collaborative websites. If you'd like to learn more about the collaboratives or interested in staying in touch, we encourage you to visit our websites and join the listservs. And then finally, while we try to minimize technological issues during these webinars, we do apologize for any that might arise. Before we get to our presentations, I'm just going to cover a few logistical and technical items. So we have muted all attendees just to avoid interruptions and background noise, and attendees will remain muted with webcams disabled throughout the webinar, but we do encourage and will accept written questions from attendees, and please use the Q&A function to submit your questions, which will be answered at the end of the presentation. And then if you do experience any technical difficulties, you can either use the chat function to fix those, or you can email Connor. Connor, his email's right on the screen. And with that, I think I've finished my beginning spiel, and we can get to what we're all here for, which is the presentation. So I'm just going to introduce our two wonderful speakers today. Dr. Anna Baig-Hold is an algal toxin and ecology research specialist with the Cooperative Institute for Great Lakes Research. Anna works with the harmful algal bloom monitoring team at NOAA's Great Lake Environmental Research Laboratory in Ann Arbor, Michigan, to study ecosystem dynamics of HABs in western Lake Erie and Saginaw Bay, Lake Huron. She is also interested in science communication and strives to connect her work with communities who are impacted by HABs and other environmental issues. Our second presenter is Vincent Denneff. He is an associate professor in ecology and evolutionary biology at the University of Michigan. His lab studies how species interactions affect the structure and functioning of microbial communities in freshwater lakes, including the Great Lakes. Species interactions of focus in current research include the impacts of consumers, including invasive mussels, and the impacts of host microbiome interactions on community dynamics. An emphasis is put on how such interactions affect phytoplankton community dynamics, including the fate of cyanobacteria harmful algal blooms. All right, and with that, I'm going to end my slide, or stop sharing my screen, and Anna, you can take over and start your presentation. Thank you.
Cool. Hi, everybody. Before I share my presentation, I just need to highlight my custom-made limited edition Detroit mussel shirt that has a quagga mussel in the middle of it. Yeah, I just thought you all, as a group, would find that as interesting as I did. But yeah, thank you for the great introduction. We're going to be talking about quagga mussel food choice at the HABs buffet. And if you think you've heard all the mussel feeding presentations, well, stay tuned because we have some cool new details on that story to share. But first, I would like to just give credit to my co-authors, Reagan Herrera at NOAA, Paul Gleishaw at NOAA, and Hank Vanderplug, who is who was at NOAA but is retired, but is still, working with mussels because why not? And then Vincent will introduce his co-authors later. Oops, here we go. So yeah, to get started, Dreissenid mussels are ecosystem engineers. I'm just going to come out and say it. Anytime they invade a freshwater body, visible changes in the water clarity can be immediately visible. On the other hand, lakes with invasive mussels might start having annual harmful algal blooms. But, any way you look at it, when a zebra or quagga mussel, both of the Dreissenid mussels that are invasive in the Great Lakes, if they are infesting a water body, they are going to engineer that ecosystem, and it will be different than before they invaded. And I like to think of this relationship between the Dreissenid mussels and the phytoplankton, or cyanobacteria and algae, is what I mean when I say phytoplankton, as a predator-prey relationship. And if you think about mussels as a predator with phytoplankton as their prey, you can really start to unravel a story of how these predators are having major impacts on trophic dynamics in their invaded ecosystems. And just broadly speaking, predators can be clumped into two kinds of groups, where they can either be specialists, and they will focus on capturing and ingesting preferred food independent of prey density. So even, they're at the buffet, their favorite food is mac and cheese. There's only a tiny bit of mac and cheese left, and the rest is just like dry lettuce. They're going to eat, if they're a specialist, they are going to find that little bit of mac and cheese that is left and eat it and ignore all the gross dry lettuce around them, even if it takes more effort to get to that mac and cheese. On the other hand, a generalist will switch prey focus when their preferred food is limited. So a generalist will be like, you know what, there's a ton of lettuce, it's right here, I'm just going to grab this and eat it. And yeah, both kind of dichotomies work in different systems. So we know that quagga mussels can be picky eaters, and they will selectively feed on preferred phytoplankton, which I have indicated with little heart shapes here, and the little heart shapes and are also interspersed with particles that mussels don't want to eat, which is symbolized by the red circle with a cross through it. And so mussels are exposed to all of these particles on a regular basis, and they can filter in the particles that they like and eat the little, heart particles that are their favorite, and either ignore or just spit out particles that they don't want. But again, how does this selective feeding fit into that predator-prey dynamic? And how does it fit into different areas of the Great Lakes? And this is where this story starts to get complicated, and why we are now at the point where we're really looking at all of these context-dependent small details to think about how invasive Dreissenid mussels react in an ecosystem. Because as we've seen in certain areas of the Great Lakes, like Lake Michigan, the invasion of quagga mussels has basically led to the disappearance of the spring diatom bloom. And Lake Michigan is really oligotrophic and doesn't have a ton of nutrients to begin with. So, they just kind of took what was left and ate it. But on the other hand, if you're familiar with Western Lake Erie was one of the first places in North America to get invaded by zebra and quagga mussels. And they engineered that ecosystem, and now we have seasonal harmful algal blooms that are dominated by a cyanobacteria species called Microcystis aeruginosa, and that has been going on for over 2 decades at this point now. So again, we know a lot about mussels, but how they react is very context dependent. So what we want to do is see how they're going to, react when they have a lot of choices. Like in a Microcystis dominated harmful algal bloom in Western Lake Erie. What a coincidence that we end up back in Western Lake Erie. So the thing about harmful algal blooms in Lake Erie is that just like, ecosystem dynamics in the Great Lakes, they're super complicated. There's a lot going on there, and we just keep learning more and more every day about these blooms as we continue to research them. But what's really cool in terms of this project is that even in a Microcystis dominated harmful algal bloom, there are still other types of algae there, other species of cyanobacteria, green algae, diatoms, cryptophytes, other stuff besides cyanobacteria that filter feeders like mussels can prey on and eat. The graph I have on this slide is from last HAB season, 2023, from one of our sites in Western Lake Erie, and these bars show the relative concentration of these different phytoplankton groups in the lake during different parts of the year. The diatom group is this blue color, and we get a diatom bloom in like mid-May. And then we see the cyanobacteria, which is in this green bar color, if you can see it, creep up in the middle of the summer, and that's the harmful algae bloom. So there's a lot of stuff for the mussels to eat in Lake Erie. And on top of that, so there's all of this diversity of phytoplankton, but even if we're just looking at Microcystis, the dominant species, there are just countless numbers of genetically unique strains of Microcystis present in that bloom. And those strains are important because different strains can produce different chemicals. Some strains have the genetic cookbook to make microcystins, which are harmful cyanotoxins that they produce. Some strains of Microcystis aeruginosa can't produce those toxins. So, these individual genetic differences within these strains can be really important into how the quagga mussel is preying on the phytoplankton. So considering all of this, that mussels and phytoplankton can be thought of as a predator-prey relationship, and when there's a Microcystis bloom, are quagga mussels being generalists or specialists? Are they spitting back out all the Microcystis? Are they eating certain strains and not others? Will they engage in prey switching once their preferred food is gone and start eating food that maybe they wouldn't have eaten if their preferred food was available? And then again, how does this all impact the bloom dynamics of Lake Erie? And how does this impact, how just the whole community of Lake Erie and impacts to the trophic structure, and algae can have huge impacts for other species down the line as well. So the study objective for the first part of what Vincent and I are talking about today was to determine feeding rates of quagga mussels who were exposed to unique strains of Microcystis aeruginosa. That's the cyanobacteria that causes the harmful algal blooms in Lake Erie every year. And we also fed them a green algae called Chlamydomonas oblonga. And that algae is a food that we know that they selectively eat. So, as I mentioned before, a lot of the interactions between mussels and phytoplankton are context dependent. So we wanted to do a lab study, but still keep that context. So we collected quagga mussels, and the Microcystis aeruginosa was also collected from Western Lake Erie throughout these NOAA GLERL, which is the Great Lakes Environmental Research Lab, master sampling stations. And the Microcystis aeruginosa strains were collected and isolated and are cultured through the Western Lake Erie Culture Collection at the University of Michigan. And I wanted to give them a shout out because they are, open to receiving orders if anybody else is looking for some unique Microcystis strains to culture. I put their website on here. And then I also have this QR code that you can snap with your phone. And when Vincent's talking, we'll make sure that you get this website in the chat eventually as well if you don't catch it on this slide. But yeah, Western Lake Erie Culture Collection, relatively new culture collection, but super cool. Lots of different strains of Microcystis. So we can do this interesting work. So what we did is we nicely asked the Western Lake Erie Culture Collection for some strains of Microcystis, and they said yes. So we cultured those. The picture on the top left of this slide here just is an image showing some flasks culturing the different Microcystis strains inside an incubator. And I'm not sure if you can see this much detail on the slide, but we have 3 different cultures growing on a shelf here, and all 3 of them kind of look different in the flask from each other, which is again just highlighting that even though it's the same species, you can have dramatically different genetic makeup of these different strains. And then, so yeah, we combined a strain of Microcystis with Chlamydomonas, put it in a put that mixture in a bucket with 5 mussels, and we let the mussels feed on that phytoplankton mixture for about 2 hours at 20 degrees Celsius. And if you're used to Fahrenheit, that's about room temperature. Yeah, I also have some images of the bucket set up in our temperature control room. We also had some control buckets that contained algae, the phytoplankton mixture without mussels, so we could see if there was any natural settling of the algae as well that wasn't due to the mussels. And then what we did to analyze these samples is we took a water sample from the buckets at the beginning of the experiment, 15 minutes after start, 30, 45, 1 hours, and 2 hours after the start of the experiment to measure their feeding rates. And we used a fluoroprobe to do this. And the image on this slide on the left here is my co-author, Paul Gleishaw, using a fluoroprobe to analyze the samples during an experiment. And the fluoroprobe can pick up, can differentiate between pigments from green algae and pigments from blue-green algae. So it was pretty cool because if you've ever done pigment extraction analysis, it takes a couple days, but this way we were able to get our results right away. So it was pretty cool to get those results in real time and see what our mussels were doing. We also measured microcystins at the beginning and end of experiment. And as I mentioned, the microcystins are a toxin produced by Microcystis. Oops, sorry. So now the fun part, now the results. We found that in every single instance, quagga mussels filtered the Chlamydomonas at much higher rates than Microcystis. These taller bars that are green are the filtration rates of Chlamydomonas, and then these, if you can see these tiny bars next to them in like a bluish color, those are the feeding rates of Microcystis. So in some cases, the animals in the experiments were just completely eating all of the Chlamydomonas and not even touching any of the Microcystis. And in other cases, we actually saw that the mussels were not ingesting, they were capturing some of the Microcystis cells and sorting them within their gills and then rejecting them. So, in some cases, the mussels just totally ignored the Microcystis, but in others, they were actually, taking it in through their siphon and saying, no, I don't like this, and then spitting it back out. And we saw evidence of this in some of our experimental buckets. The picture on the left, I tried to put a circle around all of this clumpy green stuff at the bottom of the bucket. We are calling that pseudofeces, which is what they spit back out when they reject food particles before ingesting them. And the picture on the right here is a control bucket where we just had the algae mix in to see if it settled on its own, and there was no settling without the mussels in there. And then we also had some statistical evidence of this where we saw that, like, yeah, their capture rate was higher than their ingestion rate, which again is a signal saying that they're capturing it and thinking about it and then just not eating it. Another really cool thing that we saw in this experiment was that when there were microcystins present in the experimental buckets, the mussels ate the Chlamydomonas at lower rates than when microcystins were not present. And this is pretty incredible, right? Because Chlamydomonas does not produce microcystins. So the chemicals that the Microcystis is producing is impacting how the mussels are eating another species of algae. So the key takeaways from this side of the experiments is that quagga mussel predation of phytoplankton is not random. They were not randomly selecting particles. They were choosing the green algae, and sometimes when it was totally depleted, they wouldn't even eat any of the Microcystis. So even when their preferred food was gone, they would not switch to a more abundant prey source. So from this, we're concluding that in this scenario, they were demonstrating specialist predator behavior. And what this could mean for the entire ecosystem is that if quagga mussels are just eating the stuff that they like and leaving all the Microcystis, this makes Microcystis way more competitive because they have more light, more nutrients, more space to grow. And Microcystis is becoming competitive in, a phytoplankton community where it might not necessarily be competitive if it weren't for the quagga mussels. And then also, one thing I wanted to mention is that even though in our study, microcystins, the presence of microcystins resulted in lower feeding rates for mussels, I'm not going to say that it was because of microcystins just yet. Microcystins are just one of many, many, many compounds that can be produced by cyanobacteria like Microcystis. And many of these compounds are completely undescribed. We have no idea what they are, what their effects are. So Microcystis could be another, what could be happening in our study. It could be microcystins. It could be microcystins and another unknown compound, or it just could be a completely unknown compound that's causing these effects that is just coincidentally present with microcystins. So, as with any good study, we finish on, wow, I can't wait to do this again, but this time I want to look for X, Y, Z. So now that I've explained that quagga mussels are picky eaters, Vincent is going to give us a clue as to why they might be selecting certain particles and not others.
Okay, perfect. Thank you, Anna. I'm very happy to be part of this relay presentation. Or presentation relay, I don't know how you call it. So yeah, I definitely want to credit the co-authors on my part, which are Dylan Baker, Marvin Lin, and Zachary Bergberger, who have been working in the lab over the last couple of years, doing similar experiments than the ones Anna described, to kind of gain some further insights. Also definitely want to give a shout out to Glurl and Sigler, who I've had the luck and pleasure working with, I guess, since I started at the University of Michigan. Definitely Hank Vanderploeg was critical in sort of inviting me out on a variety of projects that they were involved in and were interested in getting some more microbial perspectives too. All right, so you can do next slide, please. Thank you. All right, so as Anna said, so the initial study done at Glurl with Ziegler included about 7 strains of Microcystis, and what we did in in our side of the study, which was sort of happening in parallel, was to expand that to about 20 Lake Erie strains. What you see on what is it, the right of this slide, you see a phylogenetic tree, which kind of shows you the relationships between different Microcystis strains that have been sequenced or were sequenced at the point that this tree was built. And sort of the darker circles and sort of the outside in general of the circle, sort of the 7 strains that Anna was mentioning, and then all the green dots that are within the multicolored circle on the outside are the whole set of 20 Lake Erie strains from the Lake Erie collection that we kind of looked at. So that expanded the number a bit across slightly wider phylogenetic range within this group of organisms, and then also allowed us, give us a little bit more statistical strength to try to look at gene-trait linkages, kind of look at the variability in how these different strains are getting eaten, and what genes, what traits might potentially underpin this. Because that's one thing with these, with the Lake Erie collection, is that we have genome sequences for all of these. And in the case of microbes, as opposed to say plants or something where you can see a lot of traits just with the naked eye, to see the differences between these different Microcystis aeruginosa, the genomic sequence really allows us to at least get some clues and some inferences of what potential traits are, which can also be then further measured using chemical ecology methods, for example. All right, next slide. All right, so methods, I copied this from Anna's slide, more or less. So again, we worked with 20 strains now, but again, the experiments were feeding them either both of them or the green algae by themselves we did as well. So that way we had a internal control across all these different experiments, because there can be a bit of variability day to day in what the feeding rates might be, the mussels of the mussels. So we wanted to be able to have some level of additional control to help out with that, especially to also be able to look a bit at further dig more deeply into what Anna mentioned of sometimes it seems the green algae are fed upon less than other times, right? And the green algae alone control also gave us some means to better characterize that. Otherwise, in methods, we have slightly smaller volumes. Well, we have smaller volumes and then fewer mussels per beaker accompanying that. And a few other small differences, but that which can definitely potentially impact a bit how the feeding rate of the same strain might look in experiment 1 and 2. Next slide. All right, so again, what we saw was quite a bit of variability. What you see here are the clearance rates. That's the initial amount that's sort of removed, but not necessarily ingested. So we have those values too, and I'm zooming in on this one first. And what you can see is that there's some, like all the way on the left of this graph, right? On the x-axis basically are the different Lake Erie strains, and then the y-axis are these clearance rates. What you see in the dotted line sort of across is sort of the normalized feeding rate of the green algae alone, right? And then basically you can sort of see the rates of the Microcystis feeding, which range from basically essentially the same as the Chlamydomonas to virtually zero. Right, so a wide range, high variability in feeding rates, which, again, Anna showed variability, and also some previous reports in the past had also shown high variability, with some being eaten quite well and some not at all. Next slide. Okay, so then, as I said, we were really interested in, like, okay, if we see this variability, right, but microscopically these all look, somewhat similar, how can we pin this down a little bit? And one thing we did was we did pre-filter these cultures using a filter size to kind of make sure that the Microcystis that was there was at least within a size range that we knew mussels could eat. So that way, any variability we saw in feeding had to be due to some differences in what they tasted like, right? So what they looked like chemically, since mussels have that really that ability to really be picky about what they're eating. And so one of the things that came out, and so again, as I mentioned earlier, so for all these organisms, we have genome sequences, and from that we have predictions on which secondary metabolites they might be able to produce. And actually, if you look at the variety of different traits that we've been zooming in on, the ability to produce a wide range of different secondary metabolites is one of the most variable traits between different Microcystis. And so Greg Dick's laboratory, geomicrobiology laboratory at the University of Michigan, they've been doing a lot of work zooming in on these. And so we've been working with them to kind of cross-reference or the things we're identifying, ensuring that we're doing correctly, since they have been staring at this a lot longer than we have. And so one of them that popped out was sort of, if you look at different strains and you see them organized on the left from more susceptible to more resistant as you go up. And one of the things you see among the more resistant ones is that they have these cluster of genes that are identified to encode the genes that are able to produce N-aminopeptin, which is a modified, which is a peptide, modified peptide, basically produced by Microcystis aeruginosa. So basically, that was the first hint that we kind of have here, that this molecule might be associated with high food selectivity. And there is a little bit of work, again, as Anna mentioned, right, the chemical ecology of Microcystis is, has been touched a bit, but there's still a lot to be learned. And so there's some clues in the literature as this potentially being involved in some resistance against feeding, but there's very little hard data for that. So this is one of the first studies that kind of seems to indicate, this molecule might be one of the multiple ways that Microcystis, certain genotypes of Microcystis might be able to fend off mussels. Next slide. Another cluster that came up for several of the strains, and so some of these are similar to the the ones in the first list, but some of these are others, is that if there was a fairly complete cluster for the production of cyanopeptolin or micropeptin, these are dually annotated. Again, this is a cluster, if this is present in fairly complete form, because you can see a whole set of partial clusters as you go down the list, right, which might have some of these genes, but not the entire cluster necessary to produce this molecule. Again, this seems to be identified to be related with food selectivity of mussels against Microcystis aeruginosa. So it's basically a compound that helps protect them from feeding. Yep, next slide. All right, so so why might we care, right? Why we care about this variation in predation-resistant traits of Microcystis aeruginosa? And our lab is doing this in addition to these experiments with mussels. We're also right now in the middle of, and with weed as the royal weeds, actually, the people I mentioned earlier, Marvin, Dylan, and Zach, are, as we speak, still finishing experiments on looking at feeding resistance by different Daphnias, but Daphnia that we got from Lake Michigan, as well as some that were from McDuffie's group in our department. So, so we're also looking across those different Daphnias to kind of look at feeding resistance and kind of starting to kind of get a bit of an image. Okay, these different predators, and of course, there's many others we could include. We're kind of mussels we're using because they're so predominant in Lake Erie as a feeder, and then Daphnia because it's been studied as a model zooplankton that is quite important in shaping food webs in lakes. So we're trying to get a better image there of these different potential routes of grazing and how Microcystis defend, and how there's basically variation among different Microcystis in these defense traits. So why it's important overall to think about this is because if you actually look at, if you take predation pressure out of the picture, and this is some work that Hank, for example, was critical in the early years of the invasion, showing how with the presence of mussels was really key to the development of these blooms. Obviously, the input of nutrients is absolutely essential for the ability to bloom, but the composition of these blooms is definitely in the early years were shown to be strongly shaped by the presence of mussels, as Anna also showed with the data, by them clearing very selectively the competitors of certain Microcystis. So we've also done some experiments in the lab, basically showing that one of the reasons Microcystis needs that predation resistance and predators there to come up is because they're actually pretty poor competitors. So what you see on the graphs, basically, in figure A is P star versus N star. This is concept out of ecology, which is the minimal concentration of a certain nutrient that an organism needs to be able to have net growth. And what you can see, the algae are basically a set of green algae that we looked in the lab, and then a variety of Microcystis Lake Erie strains. And what you can see is that both P star and N star are higher for Microcystis than algae, and higher is worse because that means they need higher concentrations to be able to have growth, right? So basically, in the absence of predators, we would predict Microcystis loses competition. And what you see in the experiments below in graph B, from left to right, there's 5, 20, 80. You see there, that's the N to P ratio. And what you see is that a low N to P ratio, basically the green curves, which are is one of the green algae, easily wins compared to Microcystis, which kind of dies out. At 20, N to P of 20, you see Microcystis sort of holding on more, but then kind of falling away. And then at 80, at very high N to P ratios, you see that they kind of both are able to hold on. So basically what our data indicates, because particularly for N, so P, they're somewhat competitive. For N, they have really high N requirements, Microcystis strains. Only under really high N to P ratios is Microcystis quite competitive. But even then, it's at the margin. Now, so that means you have high N requirement for competitive abilities, but actually also for defense. So these compounds I was talking about, we identified to be involved in resistance. Those are modified peptides. So they're very high in nitrogen as well. So again, for its ability to compete for resources, right, it needs to have high N to P. For it to be able to defend well, it needs high N to P because that's the only case it's going to make these compounds. And for the ability to produce toxins, that's already been shown too, that is something that is a trait that is much more prevalent when there's high N to P levels. Now, as many of you probably know, right, our current policy is really focused on reducing phosphorus inputs in these systems with good reasons, because that's typically seen as the limiting nutrients, right? However, if that's the pure focus, right, to reduce phosphorus only, you will create a system that has higher N to P, which, at least for this particular harmful algae, right, there's others in the system as well, would mean you would actually create conditions under which these are much more able to persist and compete with other organisms in the system. So that's one implication of why to care and why to care what's involved in traits. Also from a modeling perspective, right, a variety of these trait parameters that we're measuring and the variability are things that we're working with GLERL and other collaborators to kind of think of how we could use that information to make models maybe a little bit more reflective of the complexity in the system, which is not just one organism with one set of traits, but can shift depending on the external forces. Next slide. Right, and so that's, I think, what I have for today. I think, I'll thank the people and I'll give it over to Anna again. So definitely Dylan, Yuchen, or Marvin, Zach in the lab, as well as Madeline, who's been involved also in some of the Daphnia work and the Mussel work. Definitely want to shout out to the Michigan Geomicrobiology Lab, right, which is Gregory Dick's lab, who's the director of Sigler, and a whole variety of people in their lab that we've been collaborating with over the years. Part for also access to that Western Lake Erie culture collection that came forth from that group, as well as people that we've been working with on the genomic side of things and then the secondary metabolites. Definitely also want to thank the people at GLERL, that also involved in the experiments that Anna's been doing, that have been really key in working together to get to where we are right now. Definitely, from the funding level, want a big shout out to NOAA Omics program that has supported us over the year, as well as the Michigan Sea Grant core research program for our part of the work. And I'll give it back over to Anna.
Thanks. Yeah, and then on our side, Dwayne at NOAA works and does all of the toxin analysis. If you pay attention to the weekly Erie report from GLERL, you might recognize his name. And then Glenn Carter from Sigler is one of our field leads and also a mussel researcher as well. And he went out and collected the mussels that we used for us. I also just want to say, I didn't want to spoil Vincent's talk, but so in our samples, we only measured microcystin right now, but we routinely find anabanopeptins in Lake Erie also. So yeah, I just feel like there was going to be a question if we measured that, and not as of now, but like I said, we do routinely see anabanopeptins out in Western Lake Erie. So stay tuned for more talks in the future as that data gets released. Oops, and then yeah, I think we're done. And here's another link and QR code for the Western Lake Erie culture collection. Also, please feel free to email me or get a hold of me for anything related to this talk.
Definitely the other two people I still want to give a shout out was Hank Vanderploeg that I already give a shout out to. It's been really critical in roping me into this work when I first started out, and also Regan Herrera that was wearing about 50 hats for a while there, as also Hank left, and others that has been really critical in supporting the work we've done.
Yeah, sorry, really quick more. I just wanted to let everyone, like I told Hank this, but reading Hank's mussel papers was like foundational for my PhD work. So to be like working with him on a mussel project at Laurel now is just like, it's my little science dreams are coming true. So this project's just great for so many reasons. Okay, I promise I'm done now.
No, that was great. Thank you both so much. I'm just going to take over sharing again real quick. No, let me move that over. Yeah, thank you both so much. That was wonderful. So we're going to move on to our Q&A now. And I did put that Western Lake Erie culture collection link in the chat. Looks like we have a few questions rolling in here. So the first one, I think is just general for Anna's portion of the talk. Are the results published or still in prep?
We're actually working on that right now. So hopefully my goal is to submit everything by, before the end of the year.
Awesome.
So yeah, trust me, I've been working on it and I'm really excited to get it out.
Absolutely.
And, also the expanded set, we're working on that. We don't quite know yet if we're going to make it one massive thing with also all the Daphnia work as part of it, or we have field experiments and other things. Kind of have to figure out how to partition it up, but we'll be trying to work on something very soon, hopefully submitting it later this year.
Sounds good. A question for Anna. At which concentrations did you add green algae / cyanose?
So again, I'm going to credit Hank with this. We put them in at very low concentrations. So it was about 3 micrograms per liter of chlorophyll is how we measured for both. So it's 6 milligrams of chlorophyll total, which if you're not familiar with those units, that's not a lot at all. Basically, like the low bloom threshold for Lake Erie is 30 micrograms per liter. So we were feeding them at way lower rates because we wanted to see what would happen if they ate up all of the green algae. So we wanted to have them make that choice. So we went with a really low concentration.
And we use similar concentrations too.
Okay, next question. Did any of the Microcystis cultures form colonies, or were they all present as single cells? I think Sarah Rivera gave a response. The cultures from Western Lake Erie culture collection are colonial. The colony morphology varies, and then the notes of the strains she gave a link for. They gave a link for. Do either of you have anything to add to that?
I was just going to say, Vincent mentioned they filtered there, so they were all same particle size. We did the same thing as well.
Alrighty.
Sorry, we filtered our cultures through a fine mesh sieve, so they were all under 80 microns. All the cells were under 80 microns, which is like preferable for the mussels. Yeah.
Next question. If no optimal algae is available, would there be an expected die-off or would the mussels adapt?
Well, in Lake Michigan, there are quagga mussels at like 90 meters deep, and they get like barely any food down there, and there's like a reproducing population. So mussels just in general are also known for being able to go into kind of like a stasis when conditions are bad. They can lower their metabolisms, especially if it's like super cold water. So they can adapt in many different ways. Unfortunately, we haven't seen any mussels starve to death.
Vincent, do you have anything to add or I can move on
Yeah,
to the
not
next?
really. I mean, the only thing I can think of there is like, because the cyanos only come when when the temperature become warmer. So there's a whole time in the earlier succession of the phytoplankton that they're all there's plenty to eat.
Okay, I'll move on to our next question. Kind of a lengthy one with some background. The bottom of Torch Lake outside of Traverse City at depths greater than 30 to 40 feet was found last year to be covered with quagga mussels. The lake was previously sand bottom to depths of 250 feet. What changes would you expect to the ecosystem?
I'll just start. I mean, again, it's really context dependent. So it would depend on, sorry, I'm not like super familiar with Torch Lake phytoplankton communities. But again, it could really go multiple different ways, again, like just based on what's there already, what the nutrient concentrations are, or ratios are, like Vincent mentioned. And then, yeah, just some other factors too. Sorry, I don't have a really good answer specifically for that.
Yeah, I'm not 100% sure to predict either. I think Torch Lake was invaded by zebra mussels before as well, right? So, but then there's a takeover now in more recent years by Quaggas maybe, and that explains where things go deeper. Because I think that was in Lake, from what I remember, in Lake Michigan also, and Lake Huron, like the zebra mussels kind of stayed in the near shore shallows, and then when quagga mussels came in, they're able to go just much deeper because they don't need as much substrate, hard substrate to attach to. I mean, they need something to attach it, but it seems to, a kernel of sand seems to be enough to start a cluster, it seems. So, I mean, in in Lake Michigan, when that transition happened, you had this collapse. That's when the collapse of the spring phytoplankton bloom happens, because there was just so much predation pressure, and also the quaggas seem to be much more phenotypically plastic, or like than zebra mussels, in the sense that they can, like what Anna was explaining, they can just sort of like, okay, a little bit more dormant now, there's no food, and then the food is there and boom, they kick into action. Versus zebra mussels, I think from what I remember, had a lot more die off in the winter, need time to kind of recuperate and kind of build up again, and so the spring phytoplankton bloom was more safe. So I'm not 100% sure if that's what's going on. So yeah, again, as Anna said, a lot of factors depend. There were zebras already, so and from that, there could have been small, since it's probably a lower nutrient lake, there might have been small, Microcystis blooms happening, not the size of Lake Erie, of course, but like in what you see in what it was told by A. Sarnelli at Michigan State. He said if you see Microcystis colonies in a low nutrient lake, there's mussels, because that's the only way they really are able to develop in these systems, is when all the competition is taken out. And actually, in a study that we did with A. Sarnelli at Michigan State, we actually saw that the Microcystis you find in these lower nutrient lakes are their own genotype by themselves. You actually find them in the higher nutrient lakes later in the bloom, or in somewhat lower nutrient regions, because as the bloom develops, you deplete nutrients to such levels that these low nutrient types come in, and so that they are specialized in these lower nutrient availability conditions, and actually probably contribute to prolonging the bloom. But yeah, so that's as much as I can tell.
it's hard to say too because they're two huge stressors, harmful algal blooms and invasive Dreissenid mussels in the Great Lakes. So they're like, they're both coming in and just messing everything up, to put it, not in scientific terms. But yeah, and that's kind of why I wanted to really stress some of the context dependencies, because, things could go multiple different ways. But I always think of Microcystis and mussels as being two species that are really good at establishing in like a disturbed ecosystem. So yeah, good luck with Torch Lake though. And thanks for, being a voice for one of our inland lakes.
Absolutely. Looks like we have about 5 questions left in the chat, and we got about 4 minutes left. So afterwards, if there's any left, we can always email them to Vince and Anna and get them answered. Next question is, presumably anabanopeptins are expensive to produce. Is there any evidence that there is a cue from predator presence and mussel, if you will, that triggers anabanopeptins expression?
Yeah. With my best French from my home country, say eau de moule, probably, but really good question. I think that's some of the next studies that we want to do, is kind of looking exactly at the expression, right? Is it induced? Is it, to get a better clue?
Yeah, I agree. It's a great question.
It is, and yes, confirming it is very expensive, right? It's a very nitrogen-rich compound for an organism that has very high nitrogen demands.
All right, our next question is, has similar dietary research been done on zebra mussels or any native freshwater mussels?
Zebra mussels can selectively filter too, so can unionids, which are the family of freshwater mussels in North America. And most shellfish really can do that mechanism. I'm not I don't know about zebra mussels. There's definitely diet studies on unionids, but I'm not a unionid specialist. If you email me more specifically, I can probably send you some papers to look at.
All right, next question's for Vincent. When green algae are removed through predation, would you expect that more nutrients would be available to Hyscanus, resulting in greater growth?
It's a good question. I'm not 100% sure. I would have to kind of look into. I mean, I know Hank has done quite a bit of work on excretion and the stoichiometry of the excretion products. And mussels, it depends a lot on. I know for phosphorus, a lot of work was done under low phosphorus conditions. They really hold on to that phosphorus. In their high phosphorus conditions, they release it all. I would have to look back at the end, but it's a really good question.
All right, and then next question, would you expect clearance rates to be different at variable algae concentrations, 3 versus 30, in a way of, oh, sorry, my chat is moving around here, in a way of the more food, the merrier?
Yeah, so sorry, we're going to talk about Hank again, but I think in 2009 paper, I don't know, one of Hank's papers, he talks about the incipient limiting concentration. So sometimes when there's like an overabundance of algae, they might lower their rates a little bit. And again, that's why we wanted to feed them really low concentrations of algae, because we wanted them to, deplete the preferred food to see what would happen to the Microcystis after their preferred food was gone. But yeah, that would be good experiments too, is testing different concentrations to see if there is a threshold at which this, feeding behavior isn't impacted by the Microcystis anymore.
Yeah, and I think from what I understand, up to the point that it gets to sort of limit, inhibiting feeding, there is there's definitely a certain linear response of a higher rate when there's more food of the preferred food source, as far as I understand it. I don't know to what extent it's been also looked at for Microcystis. But.
Alrighty, I think we are right at time here. We do have one more question in the chat. If either of you want to answer that, talking about, is there any plans to expand to other cyanos? And then there's a species name in there or genus name in there that I cannot pronounce. So I'll let you both take care of it. But to be mindful of folks' time, I just want to give a big thanks to our presenters and all that joined for today's webinar. As a reminder, the webinar will be is recorded and will be posted on the Invasive Mussel Collaborative and HABs Collaborative websites over the next few days. If you have any additional follow-up questions or comments, please feel free to reach out to us at the Great Lakes Commission, and we can, get your questions answered. And thank you all for coming, and I will close out the webinar.
Bye. Thanks, everybody.
Bye. Thanks, everyone.
Cool. So do we just leave now? Okay. Cool. I'm going to leave. Bye, Vincent. It was great presenting.
Bye. Thanks, Anna. This was great. Thanks for sharing this moment.
Thank you.
Yeah, have a great time up north.
Thank you.